Hi everyone, I am Dr.J.SUNDARI, Head and Assistant Professor of Botany, A.D.M.COLLEGE for Autonomous, NAGAPATTINAM. Now I am going to talk about the structure, reproduction and the life cycle of Polysiphonia. Systematic position, division Rhodophyta, class Floridiophyceae, order Xeramales, family Rhodomeliaceae, genus Polysiphonia. Occurrence of the Polysiphonia. The genus Polysiphonia is represented by more than 150 species, out of which about 16 species are reported from India. They grow in marine habitat and are cosmopolitan in distribution. Commonly, they are found in littoral, sublittoral zones. In India, they are found in western and southern coast. Commonly, they are grow of lithophytes on rocks or stones. Some species like Polysiphonia urceola, Polysiphonia volutacea grow as epiphytes and laminaria. Polysiphonia fastidicea grows as semiparasites on Ascophyllum notatum. This slide shows different types of red algae. Plant body of Polysiphonia. The plant body is multiaxial, well-branched, thallus, of dark brown or reddish or bluish red coloration, appearing as a very small bush. The height of the bush varies from of a few to several centimeters. Most of the species are heterotrichous in habit, consisting of prostate and erect system. These species are found in prostate or erect system. Prostate system, it may be multiaxial and well developed from the lower side of the prostate system, and many unicellular rhizoids are developed. The rhizoids are much lobed at the apex and from define the attachment disc. In Polysiphonia elongata and Polysiphonia violacea, our prostate is the absent and many rhizoids developed from the lowermost cells of the erect system and by arranged by the form of massive attachment disc. The erect system filaments develop from the prostate system. The erect system consists of main axis and many branches. The branches are two types, long branches and short branches. The long branches are called branches of unlimited growth or long branches, lateral branches, and the short branches are limited growth are called trichoplasts. The long branches develop in a spiral or radial symmetry. The trichoplasts are spirally arranged, dichotomously branched, colorless, and mostly annual structure bearing sex organs. The trichoplasts may develop both from main axis and long branches. The main axis and the long branches consist of central span of many elongated, cylindrical cells situated in vertical rows. It is surrounded by 4 to 20 peripheral spans. So the plant body is polysiphoneous and named as Polysiphonia. Only the central span is present at the apical region, both main axis and the long branches. In most of the species, pericentral span is covered by 3 layers of cortical cells formed due to periclinical and periclinal division of the cells, pericentral span. All the plant bodies are connected with each other by pith connection. The short branches are trichoplasts or monosiphoneous. Cell structure of Polysiphonia. The cells have thick wall differentiated into outer pectic and inner cellulosic layer. The cells are uninucleate with many discoid chromatophores without pyridones. Neighboring the cell are connected by pith connections. The cells contain large central vacuoles reserved for Floridian starch. Important features of Polysiphonia. The plant body is Polysiphonia. Apical growth takes place by single dome-shaped apical cells. The sexual reproduction is advanced euchromus type. Post-fertilization stages is much elaborate. Cystocote is well developed. Reproduction in Polysiphonia. The Polysiphonia reproduction takes place both sexual and asexual. The sexual reproduction is a euchromus type. In the life cycle of Polysiphonia, three kinds of plants recognized. There are diploid tetrasporophyte, haploid hermitophyte, and diploid carbosporophyte. Diploid tetrasporophyte, it develops on direct germination of carbospore, thus the plant is diploid. In the independent plant, which is instead of developing sex organs, develop tetrasporangia. The diploid nucleus of tetrasporangia undergoes meiosis and develops 4 tetraspores. Haploid hermitophyte. It diploids on direct germination of tetraspore, thus the independent plant is haploid. Most of the species are heterothallic, thus the spermatangia and carbophonia, and develop on different plants. And the third one, diploid carbosporophyte. This stage is diploid and dependent on haploid gametophytic plants. The union between haploid spermatangium and haploid female, they form diploid nucleus inside the carboponium for the development of diploid. The nucleus forms diploid carbosporophyte. Later carbospore are formed by mitotic division of carbosporangium. The carbospore are direct germination from diploid tetrasporophytic plants. Asexual reproduction. The asexual reproduction takes place by the haploid non-motile tetraspores. These carbospores are direct germination, developed diploid tetrasporophytic plants. The plants are independent and polycyphanous. Some pedicendal cells of the thallus near apical region develop sac-like tetrasporangia. The diploid nucleus for tetrasporangium undergoes meiosis and form 4 tetraspores. These spores are arranged tetrahedrally. This slide shows development of tetrasporophyte, different species of Polysiphonia. The tetraspore are produced in tetrasporangia. Single pedicendal cells are each tied up towards the apical region. The function of tetrasporangial initial. This initial cells is smaller than the other pedicendal cells of any particular tier. These initial cells divide vertically into inner and outer cells. The inner cells function directly into sporangial mother cells, and the outer cells further division to form 2 or more cover cells. The sporangial mother cells divided transversely into lower stalk cells and upper tetrasporangial cells. The latter undergoes further enlargement and develop into a tetrasporangium. The diploid nucleus of tetrasporangium undergoes meiosis and form 4 tetraspore and meiospore. The tetraspore are arranged tetrahedrally inside the tetrasporangium. The mature tetraspore are liberated by rupturing the wall of the sporangium. On germination of the developed the gametophytic polycyphanous plant. Being heterotetrally, out of 4 tetraspore to produce a male and 2 remaining to produce a female gametophytic plants. Sexual reproduction. Sexual reproduction takes place the ookamous types. The plant are commonly dioecious. The male sex organs, spermatogea, and the female sex organs, carboconia, are developed on male and female plant respectively. The male reproductive organs, it is called spermatogeum or anthedium. Initially, the male trichoplast develops a side branch on the plant body. It becomes a branch. In some species, both the branches becomes fertile, but the other only one remaining the fertile and the rest undergoes repeated trichotomous to form trichotomously sterile structure. The monosciponous fertile branches of male trichome bears many unicellular and spherical spermatogea. Each spermatogeum, it is a uninucleate structure. It produces a single spermatogeum, the male gamete. This slide shows development of male reproduction organs. During the development of spermatogea cells, expect a few basal cells divide periclinical and form pericentral cells on both sides. Each pericentral cell undergoes several divisions and form spermatogeal mother cells. Each one cut off 2 to 4 unicellular bodies. This spermatogeum developed in a single non-motile male gamete, the spermatogeum. The spermatogia are liberated from the spermatogeum through narrow apical slit on the wall. The spermatogia are dispersed through the water. The female reproductive sex organs is called carboconium. The carboconia develop at the top of the 2 to 4 called carboconial filaments. The carboconial filaments develop on the female trichoplast. The carboconium is a flag-shaped body with the facial swollen region containing an egg and upper elongated neck regions, the trichokines. During the development of carboconium, initially the female trichoplast initially is developed on the central chiffon, a few cells below the apical cells. The development of female reproductive sex organs. The female trichoplast initial then the undergoes repeated division and form of female trichoplast of 5 to 7 cells. The lowermost 3 cells of the female trichoplast divided vertically and form 3 tiers of pericentral cells. Any one of pericentral cells, there are many tiers towards the mother axis becomes the supporting cells. The supporting cells cut off small initial, it is outside, and the procarp initially undergoes repeated division and forms of 4 called branches. The procarp are proconial filaments. The apical cells of the proconial filaments function as the proconium mother cells. The cells further develop into a carboconium. The carboconium has a swollen facial region containing egg and elongated tubular region of the trichokine. At the later stage, the carboconium develops 2 initial to supporting cells, one at the basal and the sterile filaments, initial and other to the later stage, the later sterile filament initial. The later sterile initial dividers transfer and form these 2 called later sterile filaments. The carboconium is ready for the fertilization at this stage. The pericentral cells adjacent to the supporting cells start growing to cover the fertilized carboconium. Later on, they form sheaths around the fruit body called procarps. Fertilization. The spermatogia are dispersed with the help of water. A few spermatogia becomes attached at the tip of the receptive trichogyne. Out of many, only one becomes successful. The common wall of the successful spermatangium and trichogyne dissolve at the point of the contact and the male nucleus passes to the female nucleus present at the face of the carboconium. The fusion between the nuclei result in the formation of zygote. Post-fertilization stages. At the starting of this phase, the auxiliary cell is developed from the supporting cell just below the facial region of the carboconium. Simultaneously, the later sterile filaments increase in the length of bi-cell divisions as well as elongated and facial sterile initial divides to form a two gall filaments. The auxiliary cells has a single haploid nucleus. This slide shows Polysiphonia species. The tubular connection with the developed between auxiliary cell and carboconium. The carboconial nucleus divides mitotically into two nucleus, which one is transported the auxiliary cell and other than the remaining in the carboconium. Thus, the auxiliary cells contains one haploid and one migrated diploid nucleus. The haploid nucleus is degenerated gradually to the trichogyne stivers. Many vegetative filaments then develop from the adjacent vegetative pericentral cells, which gradually develop to the total covering. The diploid nucleus of the auxiliary cells then divide mitotically and form two nucleus. One of them migrates into the outgrowth developed on the auxiliary cells. This outgrowth of this separating by the wall formation, the conioplast initial. In this way, many conioplast initial can develop on axillary cells, each initial by repeated mitotic division to form conioplast filaments. The terminal cells of the conioplast filaments develop into a carboconium which form single diploid carbospore inside. During this development, the auxiliary cells, supporting cells, carboconium and some cells of facial and sterile filaments fuses together and form a irregular cells in the placental cells. The haploid cells, the haploid nucleus of the pericentral cells gradually generated and have simply a duty functions. The placental cells, conioplast, filaments and carboconia are covered by many vegetative filaments and form an URNA-shaped structure, the carbocystocard. The outer covering of cystocard is called pericard. The diploid part of the cystocard represent the carbosporophyte. Some cells of facial and sterile filaments along with some cells of carboconial filaments gradually degenerate. The carbosporangium develops single diploid carbospores after liberating from the carboconium, comes out through the ostial or cystocard. This slide shows Polysiphonia species, the branch period in the cystocard. Germination of carbospore. Coming in connection with any solid surface, the diploid carbospore gets attached, then undergoes first mitotic division and form large upper and small cells. Both the cells undergoes mitotic division and form four-call stage. The lowermost cell forms the rhizoids and the uppermost one functional apical cells. The rest of the cells undergoes further development and form the Polysiphonia body. This plant body is diploid. The tetrasporophytic plant which later develops and the tetraspore and complete the cycle. Life cycle of Polysiphonia. The life cycle of Polysiphonia consists of three distinct phases. The diploid tetrasporophyte and the haploid gametophyte and diploid carbosporophyte. Out of four tetraspore produced in tetrasporangia on diploid, the tetrasporophytic plant to tetraspore developed haploid gametophytic male and other two haploid gametophytic female plants. The male gametophytic plants developed the male gametangia inside the spermatangia and the female gametophytic plant developed female gametes inside the harboconium. The zygote developed inside the harboconium after gamete fusion with the gradual development of conium plus filament. Carboconia and carbospore are developed inside the cosmocyte structure, the cystocar. In its carbosporophytic state, the diploid carbospore on germination produce the diploid tetrasporophytic plant against. Thus, the life cycle is triphasic and haplodiploid type. Thank you, Mananta.